Okay, so let's just start with the scale of this problem, because we're not just talking about data in some abstract sense.
Not at all.
We are talking about, what, millions of individual cells? Each one is like a needle in a haystack of 20,000 genes.
Exactly. Single-cell RNA sequencing has been this amazing engine for biology. But the data it produces, these massive count matrices, have created a silent bottleneck,
A bioinformatic bottleneck.
a huge one. For years, if you wanted to get real insights, you needed custom codes, specialized software, and frankly, you needed an expert bioinformatician on speed dial.
But what if you could just skip all of that? What if you could just talk to the data?
And that's the big idea.
Today, we are diving deep into CellWhisperer. It's a new AI model that lets researchers interrogate all this complex data using just natural language chat.
It's a pretty groundbreaking concept.
Yeah. So our mission for this deep dive is to really unpack how this multimodal AI actually works. Look at its frankly surprising ability to predict things it's never seen before, and compare its speed to the old way of doing things. Okay, let's unpack this.
Right, and CellWhisperer is such a critical shift because until now, the sheer difficulty of the analysis was, well, it was fundamentally limiting the pace of science itself.
Right.
scRNA-seq is essential. It lets you untangle all the different cell types in a sample, which is the core goal of huge projects like the Human Cell Atlas.
But the data itself is the barrier, these count matrices you mentioned.
Exactly. It's just rows of genes, columns of cells filled with numbers. To pull any meaning out of that, you need to know how to run clustering algorithms, differential expression. It's a very specialized craft.
It's a steep learning curve that basically keeps the biology locked behind a programming language.
Precisely. And CellWhisperer's whole promise is to just shatter that barrier. It lets you explore the data by asking questions in plain English.
No special syntax or anything?
Nope. No complicated format needed. The interactivity is the key. You're moving from just looking at a static data dump to having a real-time conversation.
So what kind of questions can a biologist actually ask it?
Really intuitive ones. Like you could select a cluster of cells on a chart and just ask, what are these selected cells?
Okay.
Or you can ask something that needs to cross-reference your data with published knowledge, like what is the role of the KLRD1 gene in natural killer cells?
And the system doesn't just give you a textbook definition, right?
No, and this is crucial. The answers combine the data from the cells you actually selected, their specific gene expression, with the LLM's vast biological knowledge.
So if you ask about natural killer cells, it's analyzing your cells in your data set.
Correct. The system might come back and say, yes, these look like NK cells because they highly express genes like NKG7, KLRD1, GZMA, and it connects those specific expression values to the known function of those cells.
That's a level of synthesis that, that used to take a biologist and a bioinformatician working together for hours, probably days.
Easily.
So how did they do it? How do you teach an AI to translate raw gene expression numbers into, human language? It feels like they had to build a Rosetta Stone for biology.
That's a great way to put it. The whole architecture is a triumph of what's called multimodal learning. It's basically two AI models that are woven together, and they built it in 3 big steps, starting with just a massive amount of data.
Okay, so step 1 was the data curation. They had to create the raw material. The source says over 1 million matched pairs of RNA profiles and text descriptions. That sounds impossible to do by hand.
It was done semi-automatically. They actually used other large language models, like Mixtral, to help them clean up and standardize all the messy metadata you find in public databases like GEO and Solveig Gene.
So they used an AI to prep the food for another AI.
That's a good way of thinking about it, yes. The LLMs helped create clean, standardized text summaries for each complex genomic profile.
Okay, hold on, I have to interrupt there. If they're using one LLM, Mixtral, to standardize the data for the CellWhisperer model, isn't there a risk of, baking in some kind of bias from the start just based on whatever Mixtral was already trained on?
That is a very sharp and very relevant critique, and it's a necessary trade-off you make for scale. It's definitely possible they introduced some subtle biases, but the standardization was critical.
Why?
Because the metadata in those databases is a mess. One lab calls a cell T-cell subtype A, another calls it CD8 plus cytotoxic. Using an LLM helped create that single coherent language they needed to train the next step.
Okay, that makes sense. An unavoidable shortcut, but good to be aware of. So this is where it gets really interesting. Step 2. The embedding model. This is the core of it, the brain.
Right. The best analogy is the CLIP model, which is famous for connecting images and text.
Yeah.
CellWhisperer basically adapted that same idea, but to connect RNA profiles and biological text.
So instead of a picture of a cat and the word cat, it's a gene profile of a T cell and the description T cell.
Exactly. And to do this, it uses two specialized processors. It uses Geniformer for the gene expression data and BioBird for all the biomedical text.
So one part of the brain only reads the genetic language and the other part only reads the scientific literature.
Hmm. And the whole goal is to train the model to place the transcriptome vector, that's the raw data, and the text effector, the description, right next to each other in this huge 2048-dimensional joint embedding space.
Okay, a 2048-dimensional space sounds incredibly abstract. What's a simple way to think about that joint embedding space?
Think of it like a giant, detailed map of all biological concepts. If you have the data for a T cell, and you have the written words T cell, the model learns to put those two things on the exact same spot on the map.
I see.
They become mathematically interchangeable. The AI isn't just memorizing definitions, it's learning a kind of scientific abstraction.
So it's learned this unified language of biology.
And this gives you the CellWhisperer score. It's a number that literally quantifies how well a free text query like the word infection matches any given cell profile in your data.
Okay, so that embedding model gives us the score. It tells us how well things match. But how do we get to the actual conversation, the chat experience?
That brings us to step 3, the chat model. For this, they customized an open-source LLM, Mistral 7B, and they fine-tuned it to incorporate those CellWhisperer embeddings. This is what makes the chat transcriptome aware.
Ah, so it's like how some models can see an image you upload and talk about it.
It's the exact same principle. You're basically uploading the gene data as the image, and the Mistral-based chat model can now discuss it intelligently because of that powerful embedding engine underneath.
So we have the engineering, but the real test is performance. How did this thing actually do when they benchmarked it?
The results were genuinely surprising, mostly because of its power for zero-shot prediction.
Meaning it could do things it wasn't explicitly trained to do.
Exactly. It was never trained on a list of categories like predict if this is a B cell or a T cell. That ability just emerged from learning the general connection between the RNA data and the biological text.
Which suggests it's not just memorizing, it's deriving actual biological rules.
Right. And the predictive power was really robust. In the huge Tabula sapiens dataset, it achieved an AUROC value of 0.94 at distinguishing 20 common cell types.
An AUROC of 0.94 that's a stunning number. But in practice, for a researcher, what does that mean?
It means it's reliable. It means if the model tells you a cell is a T cell, you can trust that classification almost perfectly. Far, far better than random chance.
And it wasn't just cell types, right?
No. It got an EROC of 0.82 for predicting 229 different disease subtypes, like telling apart different cancer profiles, and an EROC of 0.87 for predicting which tissue the cell came from.
So with high reliability, it can tell you the cell type, the disease state, and the organ of origin, all without being specifically trained for those tasks. That's really the definition of emergent intelligence.
And here's the kicker. The model actually performed better than a widely used standard marker-based method. And it was on par with other specialized AI models that were fine-tuned specifically for those tasks.
Which really validates the whole approach. Okay, let's go beyond benchmarks to actual discovery, because this is where the speed comes in. The sources highlight a really complex study on human embryonic development.
Yes, a data set spanning from 3 to 38 days after fertilization. It's messy, it's heterogeneous, and the breakthrough was that you could get deep insights from incredibly simple queries.
Like what?
Just typing the word heart into the chat was enough to start identifying different developmental stages and the gene patterns associated with the cardiovascular system forming.
If I could jump straight to analyzing gene activation and heart development just by typing heart, that would save months of prep work. That's the game changer.
Absolutely. The analysis ended up identifying new marker genes for 10 organs that were strongly supported by the literature. So it's not just labeling what we already know, it's pointing to new, validated biological signposts.
The sources gave a perfect example of this workflow with an inflammatory bowel disease dataset. Tell us about that speed comparison.
Right. The software is integrated with the CellCouch Gene Explorer, so you can move between looking at the visuals and just typing commands. A researcher typed in the query, "Show me stem cells."
Just like that.
Simple as that. And CellWhisperer instantly identified a specific subset of well-known epithelial stem cells and, more importantly, correctly found that they were less common in the inflamed tissue samples.
Which is a clinically relevant finding.
A non-trivial one, yes.
So contrast that instant answer with what a bioinformatician would have had to do to get to that same conclusion the old way.
Well, to reproduce that exact same finding, identifying the cells, correcting for batch effects, annotation, analyzing the prevalence, that required a full conventional bioinformatics pipeline.
And in practical terms, what does that pipeline look like?
The sources estimate it required about 400 lines of custom Python code. It needed 5 different specialized software tools, and of course, the time and expertise of an experienced bioinformatician. We're talking days, maybe weeks, of focused work.
A single chat query versus weeks of expert scripting. The difference in accessibility is just stark.
It is, and it brings up an important point. CellWhisperer doesn't replace that deep, complex analysis, but it acts as this incredibly rapid first-pass tool for exploration and for generating hypotheses.
It lets the biologists get their hands on their own data immediately.
Right. They can rapidly test ideas before they commit to weeks of coding.
So if we step back, what this all means is that CellWhisperer is drastically lowering the barrier to entry. It's making bioinformatics much more intuitive, much more conversational.
It is, but we do have to include the caveats. The researchers are very clear. This is an exploratory tool. Any results still have to be validated by conventional methods before you publish.
Of course, it's still an AI, and like any LLM, it has limitations.
Big ones. First, it can hallucinate. Because it's built on language models, it can sometimes just make up overly specific details. It might say something like, these are T cells from an 85-year-old male, when that specific demographic info isn't even in the data.
It prioritizes sounding fluent over being factually accurate, a classic LLM problem.
And second, it can only be as good as its training data. Areas of biology that are not well represented in those public databases, rare diseases, understudied populations, CellWhisperer won't model those well. Garbage in, garbage out.
Right. But even with those limitations, what CellWhisperer is doing is emulating a high-speed conversation between a biologist and an expert bioinformatician. It's turning data into a library you can browse with plain English.
And if you connect this to the bigger picture, it's really proof that natural language is becoming this human interpretable integration layer for science, not just for single cells.
You mean across different scales of biology?
Exactly. You can imagine this kind of model connecting AI systems that understand molecules, cells, organs, even whole individuals, and letting them all sort of share perspectives on the same scientific question.
A shared language for different AIs.
And you should really consider what other scientific fields could be revolutionized by this, from neuroscience to particle physics. What happens when you can make any complex data instantly conversational?
When any expert analysis can become an interactive dialogue, a truly conversational lab assistant. Now that is a future worth exploring.
It really is.
We'll leave you to ponder that thought until our next deep dive.